All right, so as I'm recording this on December 15th of 2020, the Public Health England has discovered this new strain of this new variant of the SARS-CoV-2 virus, and I've seen some news sites talking about this a little bit, and so this new this new variant has this aspartate at the 501st amino acid position mutated into a tyrosine. And what's interesting about this is that it's, as opposed to a lot of the other mutations that have been discovered in SARS-CoV-2, is that this is a mutation in the receptor binding motif of the spike protein that the virus uses to bind to the human ACE2 receptor. So this is in a place that is important for the virus actually infecting human cells, and it's also sort of the part of the spike protein that our that our vaccines are targeting, and so people seem to be interested or maybe even fearful that this new variant could sort of render our vaccines sort of useless. And so I wanted to go over a little bit about viral and SARS-CoV-2 in particular, replication and mutation. Um, and so mutation in SARS-CoV-2 is not that uncommon. In fact, I have a website here, and this is actually only as late as June of this year, and this shows the mutation rate of, or it shows the phylogenetic tree of the SARS-CoV-2 virus. And so all these dots are new variants of the virus. And this is, like I said, back in June, so there would be even more of them now. So we see back here in December of last year, December of 2019, there would be a single viral variant. But then very quickly, as it started spreading, started mutating. But as I said, what is interesting about this one is that this mutation is in sort of an important place on the spike protein, and the PHE is sort of blaming this new variant uh, on sort of a spike in cases in southeast England. And so I wanted to go over just a little bit here, a little review, and I went over this in a lot more detail in a previous video I did on the Pfizer vaccine, the Pfizer mRNA vaccine. But anyway, so this is just another phylogenetic tree. It's showing a lot fewer variants here, but these are the SARS-CoV-2, just a few SARS-CoV-2 variants, and we can see they're closely related on this phylogenetic tree. And then this is the Middle East Respiratory Syndrome virus, the MERS, and we see that was split off earlier on from the SARS-CoV-2, and both of those split off even earlier on from the first SARS from the early 2000s. And so the SARS-CoV-2 uses this mRNA genome here. And like I said in the previous video, I went over more detail about the various open reading frames and so forth on it. But what I'm just going to point out here is that it's essentially their entire genome, the SARS-CoV-2 virus, the entire genome is a single long mRNA. And different parts of it actually code for different proteins, and the entire thing will be translated, and then the different proteins will sort of be spliced away or cut away from each other to become their own proteins. And over here is just the genomic organization of other coronaviruses. So we have SARS here. These other four here are all coronaviruses that can cause the common cold. And so we see that the organization of their genome is very similar between SARS-CoV-2, or this might just be the first SARS, but either way, the genomic organization is very similar. And so down here, once again, this is um, that genomic organization, and this here is what I was talking about earlier, where the entire mRNA is translated into this large polyprotein. But then some of the proteins that the virus codes for, namely the papain-like protease and the 3CL protease, will actually cut this into different sections, into the various proteins that make up the virus. And so down here is just a little illustration about how viral infection works. So the SARS-CoV-2 enters the cell. It releases its mRNA, or its RNA genome. Human ribosomes come and actually translate proteins off of it. And so these proteins can then go and then after being split apart into separate proteins. So these two things here are just the different open reading frames that the viral genome can take. So these are cut into their separate proteins, including the replicase protein, so the RNA-dependent RNA polymerase, which would be this NSP, or non-structural protein 12 and 13, as well as a few other ones like NSP 7 and 8. And so these replicases can then go and start and bind onto the viral genome and actually start producing more of these RNAs. So it can actually start producing more of its own genome, even as these are also producing the other proteins that are required, the structural proteins and the spike protein and so forth. And so this happens. A lot of the proteins are made in the endoplasmic reticulum. They can get moved to the Golgi apparatus, and then vesicles can come off of it and form the actual viral particle. So the genome and all the structural proteins come together and reform another viral particle, and in fact form a lot more. So you get, hundreds, even thousands from a single viral particle, and then that is released out to infect more cells. And so this figure here also shows some of the sorts of antiviral medications here, and where in this process these are actually inhibiting the viral replication and infection. So we have some here that actually inhibit the entrance of the viral particle into the cell. Over here we have ones that inhibit replication, and you'll notice that remdesivir, which has been talked about quite a bit, actually inhibits viral replication. And then we have these other ones down here which sort of inhibit the shuttling of vesicles between the endoplasmic reticulum and the Golgi apparatus. So down here is just a little bit more detail on how the virus actually gets into the cell. So we have the spike protein here, and this ACE2 protein here. So this right here is the human cell, and this up here is the viral cell. so the membranes of the viral and human cells. And we see that this spike protein, uh, it binds to the ACE2, one of the subunits on the spike protein. So it's what's called a homotrimer. So it's actually three proteins coming together to make a three protein complex, and one of them has this motion here, this conformational change from down to up, which lets it bind to the ACE2 receptor, and then another protein comes in and actually cuts part of the spike protein away, and that happens here at this furin site. So it will actually cleave off part of the spike protein, which causes this large conformational change, allowing it to actually bind onto the membrane of the human cell, and then it'll start bending and pulling them together so that the two membranes come together, and that's what allows it to enter into the cell. And so this process here with the spike protein is why they are interested in this particular mutation. And I have the structure here. So up here in this sort of salmon color is the SARS-CoV-2 spike protein. Down here in yellow is the ACE2 receptor. And cyan is another protein that the ACE2 receptor can bind with, but that's not important for this. And right here where I have the overlap, and sort of the purple is the part of the spike protein that binds to the ACE2 receptor. And so I actually got this from two different structures. So the purple part is actually part of the ACE2 structure where they got a little bit of the spike protein bound to it. And then the salmon overlap is actually from this ACE2 receptor structure here. So I just used those two to overlap with each other so that I could show this part here with the spike protein actually bound to the ACE2 receptor here. And so the mutation site is actually at these. So as I said, this is a homotrimer, so there's three of them. There's one here, and then we can see the one here in the binding site. So this is the amino acid residue. This I think I called it an aspartate before, but that's an asparagine. And this new mutant is mutated into a tyrosine residue. And so we can see that that is occurring right here at the interface between the ACE2 receptor and the spike protein. And so that is why they're interested in this particular mutation. So anyway, if we go over here and start talking about how mutations actually occur, so anybody who knows at least a little bit about evolution knows that in evolution what happens is you have an organism, it undergoes some kind of mutation, and that mutation can either be beneficial, which means that that this allows that organism to survive better, and therefore this will propagate further into the species, into the population. It could be neutral, which just means it doesn't make it any worse or better, or it could be detrimental, and then that particular organism would die off and it would prevent this from propagating further into the population. But this propagation here is where natural selection comes in. So natural selection will sort of, the environment will be such that organisms with this beneficial trait will thrive and be able to reproduce, and that's what allows it to propagate through the population. But the first thing we needed before we get to the natural selection part is uh, some change, some way that the organism's genome has changed, and that could be from a mutation. And mutation, I think, gets sort of a bad rep because when we think of mutants, we think of things that are, uh, well, maybe either things that are horribly disfigured, or we think of like the X-Men or something. And so we think of it as this sort of large, sort of dramatic change in something. But in biology, when we talk about a mutation, it is usually just a single base in the DNA, which can lead to a single change in a residue of our protein. So, for instance, this new strain has this asparagine here mutated into a tyrosine. And so down here, I just have sort of uh, a little overview of replication. So this is using a DNA polymerase, but it would be similar for the virus using an RNA polymerase. And so the polymerase moves along, incorporating these bases into the growing chain of DNA or RNA in the case of viruses. But you can have a wrong base come in, and that will create this bump here that will stop the elongation process. And this is called proofreading. And so there are multiple ways that organisms can proofread it. So the polymerase itself has some proofreading. So if it's moving along the DNA and it incorporates the wrong thing, this creates sort of a misshapen DNA structure. And so something on the polymerase will actually detect that, and it can actually back up, cutting off some of the other bases, and then start moving forward again, incorporating the correct bases into it. But this process essentially is not perfect. And so you will not have perfect fidelity in incorporation of these bases into the genome. And here I just have this figure that's comparing the genome size to mutation rate per site per year. And so it's looking at, if we have our DNA here, it's essentially just looking at a single site and saying that it'll mutate this many times at this site every year. And so, we can see that that isn't very much. So even these RNA viruses, which include SARS-CoV-2, 10 to the negative 2, which is 0.01, or over here 0.001 at 10 to the negative 3, which is kind of about where our RNA viruses are located. So every year at a single position, there is a mutation. There's essentially a 1 in 1,000 chance that it will mutate in a single year. But when we have thousands, millions, or even billions of base pairs, then you can imagine that could be quite often. But we see here as genome size increases, the mutation rate at a site per year actually decreases. So here with eukaryotes, with things like humans, it's down quite far from where these viruses are, and that's why these viruses can actually mutate quite quickly. Um, and so, like I was talking about, in viruses, they instead of a DNA polymerase, they use an RNA polymerase, or what's often called an RNA-dependent RNA polymerase. And so I found a paper that was looking at the proteins involved in the SARS-CoV-2 uh, replication process, and that is this NSP13 with the NSP12, NSP7, and NSP8. And this here is just some kinetic parameters for it, and we see that the turnover rate is hovering around maybe 13 turnovers per second. So essentially 13 bases is incorporated into the growing chain every second. Um, and so we can actually look at the structure for this right here. And so, as I said, it's made up of the NSP 12 and 13 and the NSP 7 and 8. And on this, we can see over here this DNA that I have in stick structures here. And this is the DNA that has already been replicated. So this is the chain of it coming out the back of this protein structure here. And we can see I have up here in this sort of ruby color, that is the NSP13, and that acts as a helicase. So there would be downstream DNA coming out of here, and this NSP13 would actually be sort of unwinding it and removing any kind of secondary structure from it, removing RNA binding proteins from it, so that the single-stranded RNA could then work its way into here, and what will come out the other side is a double-stranded RNA, because one of them is a template strand, and then the other one is actually what's being created, or what's being generated using that template strand to bind the different bases, or yeah, to base pair them, and that's how it actually knows which to incorporate, which base to incorporate. And so I have up here the NSP12, which is the actual protein that's doing the polymerase action. And so I can remove some of this other stuff here. I can move the NSP13. I can remove the NSP7 and NSP8. And we see here, this is the NSP12. And I have it colored based on the different domains that this paper was able to single out. And so there's this down here in this sort of teal color is what's called the let me look at my notes here, the nucleotidyltransferase domain, and it has an unknown kinase slash AMPase activity, and so we can actually see inside it here. There's this ADP, this adenosine diphosphate with a magnesium ion here, but they're still not sure exactly what this domain is doing, but it does seem to be able to take ADP and actually bind an AMP onto some other protein, onto a serine, threonine, or tyrosine of some other protein. And then there is the interface domain that I have up here in this sort of, I don't know, orangish color. And that, I think, is what is uh, interfacing with, well, I I don't know what it's interfacing with, but it's just called the interface domain. But what more interest here is this uh, right here called the finger domain, and they call it the finger domain because I mean that's kind of common in proteins to give them domains called fingers and think about them like hands. And you can see in purple here is what they call the thumb domain. And here in this sort of lime color is the finger domain. You can see how that sort of looks like it's a hand grabbing onto the DNA or RNA here, where this is the thumb and this is sort of the fingers grabbing over onto the RNA here. So that is the actual polymerase. And I have down here in cyan, so this base that is in cyan, this is actually the remdesivir molecule. And so we can see how that is sort of sitting right here where another base is supposed to be added. And it's sort of just sitting in that spot and it's not able to be added onto the growing chain. And so it's actually uh, inhibiting this polymerase from adding anything else onto the chain, onto the DNA or the RNA. I keep calling it DNA, but it's RNA in this virus. It'd be DNA in almost all other organisms, but in viruses it's RNA. So this is preventing the RNA from growing, this RNA chain from being polymerized. And so I actually have a figure here that kind of shows the same thing. So we have uh, the template RNA up here, and then the primer down here, and we can see this remdesivir here, which we can see is different um, from the other bases because it has this cyanide group on it, and it's not poisonous because of the cyanide. It's just having that group on here means that you can't add another base onto here. And so remdesivir, when you take it as a medication, actually comes in this form here. I'm not going to go through this whole mechanism, but it ends up as in the body as this triphosphate version, and then the pyrophosphate will be removed when it is incorporated onto the growing RNA strand, and then sort of stops that RNA strand from being able to grow any further. Uh, but yeah, anyway, that's I just wanted to talk a little bit about sort of the actual replication process, uh, replication process for the SARS-CoV-2 virus. So it uses this large complex here. It has two NSP13s bound to it, and it has two NSP. So I think the blue ones were 8. Yeah, the blue ones are 8. And we can see how those are. Oh, there's 2 of them there, and we can see how those are associating with the rest of this complex here. And by the way, I have all the papers and linked to in the description down below. So if you are interested more in things like the SARS-CoV-2 mutations, I have all these different papers linked to in the description down below. So if you are more interested in reading more about all of this, so this is the paper where I actually got the structure for the RNA dependent RNA polymerase from. This is where I got that figure from. This is the one where the structure actually had the remdesivir in the structure for it. Uh, Yeah, and so I think that is what I wanted to talk about here. Um, but I think maybe a takeaway message is uh, when you hear about like mutants, about different RNA or different SARS-CoV-2 variants, so variants or strains or mutants, these are all kind of used interchangeably. Um, there are, as I said already, just tons of different mutants, even as far back as June of this year. There are already just tons of different mutations found in the SARS-CoV-2 virus. So you shouldn't get too frightened when people start saying, "Oh, there's a new variant or a new strain or there's a mutant version of it," because that's not necessarily going to mean that it's more dangerous to humans or that it becomes immune to our 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 medications or vaccines or anything. And in fact, as I said, mutations could be most of the time. So the vast majority are going to be neutral. Some of them could be detrimental, but you probably wouldn't find that infecting people because if it's detrimental, that sort of by definition means that it's not working properly and therefore is not going to be infecting people. But it's possible, so for a small number of them to have some beneficial, and beneficial meaning beneficial to the virus. Uh, so it would benefit the virus, meaning that it would be more resistant to our medications, or maybe it would be more infectious or something like that, which is what this new strain that is being talked about here. They think, uh, they believe that this is increasing the cases seen in Kent and London, so in southeast England. They think that this new mutation could possibly be making this virus more infectious. But as I said, the takeaway message is don't get too frightened whenever you hear people start talking about this kind of stuff, because uh, most of the time that is going to be a neutral mutation and not really change the virus in any particular way. So anyway, I hope this video was at least somewhat enlightening. Uh, if you are interested in this kind of stuff, I do have videos talking about DNA replication and transcription and translation and things like that. And you can check out those videos. And as I said, if you're interested in this stuff up here, so the SARS-CoV-2 genome organization and the way that it actually replicates or translates itself and so forth, my video on the Pfizer um, vaccine, I go into a lot more detail on this. So anyway, I will see you in another video.